Hi, I am Mrs. Sloan, and this video is intended for my AP biologists. We are discussing Chapter 14, Biotechnology and Genomics. And we have already done Part 1. And Part 1, we focused on what you're seeing right here from the College Board, these processes of recombinant DNA technology, PCR, gel electrophoresis, and some of the biotechnology products that can be generated. But now here in Part 2, let me just scroll down through this presentation, which you can have access. Speaking of that, if you're new to my videos, down in the descriptor of this video, are the notes that I use for my students. And sorry, putting in presentation mode here. The notes that my students use, and column 1 is a scaffolding that they fill in, and I help them fill those in. And then column 2, I encourage them to put in pictures and images that help them with the learning of this material. So we are on Part 2, Genomics, the branch of biology concerned with the structure, function, evolution, that's all AP right there, and mapping of genomes, which is specifically asked for. So when we look at sequenced genomes, when I first started teaching in 1989, which I know you probably weren't even born yet, but when I first started teaching, they were first discussing mapping the human genome, and the techniques and the procedures. The cost of it has gone down so much, and the speed of it has increased so much that it's now becoming more common, this process of mapping out these genomes. And what is it that we can learn from it? What can we do with that? So on your notes, it's 14.4 in the notes, genomics, defined as the study, and I'm going to jump back one page just to help you with this so you can augment it a little bit. The study of our complete genetic makeup and that of other organisms and that of other organisms. So what are we doing? That structure, function, evolution, mapping. Okay, we have, you and I, about 3 billion base pairs. Remember ATs and GCs, those are base pairs in the human genome, and that includes the ones that are actually, that includes information that can actually be transcribed and translated, and also all of those non-coding sequences and what do they do. So we're going to talk about intergenic. Those are things outside of the gene, in between the genes, and intragenic within genes like introns. We've talked a little bit about that already. Sorry, gotta take off my sweatshirt. And what's known, what's unknown to us at this point. All right, so sequencing the human genome. So here, people start to make comparisons between how much of the DNA, let's say between you and I and a chimp, how much of it is similar, and then you can look at structurally similarities. You can study genomic differences between different groups of people, and that's called the HapMap project that's going on, and looking for relatedness in disease and the treatment of that disease, and that's leading towards like this idea of personalized medicine, what works specifically for you and your individual genome. So I gave you those notes on there. The Human Genome Project took 13 years to sequence the human genome. Technology has improved over time. Now 2 million base pairs of DNA can be sequenced in about 24 hours. Let's talk about differences between us. Those are called polymorphisms. And this is going to be underneath the banner of the structure of DNA, that aspect of genomics. And so polymorphisms, stick myself over here maybe, are small regions of the DNA that can vary between individuals. Most of those are SNPs, or single nucleotide polymorphisms, just one difference in one base. Most have no effect, but then others can cause disease. So on your polymorphisms, you want to have in your notes small regions of DNA that may vary between individuals. And I gave you the rest of that. Functional genomics is working backwards. It's looking to see, okay, so in your notes, now functional genomics, working backwards from the RNA. Remember who codes for the RNA? The DNA, right? So working backwards from the RNA to identify genes that are expressed. So why are these SNPs so important? It's where that polymorphism is. Is it in between genes? Like you could have gene A here and gene 0 over here, whatever. You could have it not in the gene itself. If it's within the gene itself, it's called intragenic. If it's between the genes, it's intergenic. And so that's why it could have a big difference or not so big of a difference. So in general, underneath structure of the eukaryotic chromosome, in general, more complex organisms have more complex genes with more and larger introns. And then I gave you the definition right there. Intragenic sequences are within a gene itself, and 95% plus of the average human gene is just introns. I think I have a good picture coming up here. Oh, so here, I can help you with that. Okay, so if you look right here, the genes are in purple, okay, and then intergenic in between the genes, right, are these yellow areas here. So now when we make our mRNA, right, which you can see right here, within this gene, so here's this purple gene, we're bringing out this purple gene, you can see within the gene itself, we have those introns, right? The introns are what you would call intragenic within the gene. Okay, so introns are intragenic within this purple gene right here are these green regions that you could cut out, right? And then in between genes, that's intergenic. Okay, so introns are now considered, these introns right here are now considered as regulators of gene expression, So here you can look at the complete breakdown of our DNA. Look right here at the top. Remember, exons are what get expressed. Only about 1%. So think of the chromosome itself and all those base pairs. Only about 1% is expressed. Remember what transposons are? Those are jumping genes, right? That would be an example of intergenic. You can have large duplications. You can have simple repeats. All of these, okay, all of this over here to the right, that would all be considered intergenic. Intragenic is this 24% right here, these introns, and that these can be considered regulatory. These out here could be failed attempts at evolution. This could be our DNA migrating around. Eukaryotes are not really good at keeping a clean house as far as our genome is concerned, whereas bacterial cells are better about that. So on your notes, intergenic sequences defined as DNA sequences that occur between genes. That's in your notes. The types, and I'm listing them right here, repetitive DNA elements, very common, 2 or more nucleotides repeated many times. That can be up to half of the human genome. You can have tandem repeats, sequences which are next to each other on a chromosome. Think STIRs, those short tandem repeats. Interspersed repeats placed intermittently along or among chromosomes. Examples, it may play a role in the evolution of new genes. Transposons are jumping genes, ability to move within and between chromosomes. They can sometimes also be regulatory, depending on where they jump into. And then there are unique non-coding DNA may actually create small-sized RNAs that are more effective than proteins at times. Remember when we talked about the small nuclear RNAs? All right, so I think probably what I would take away from this, okay, particular information is when you look at our genome, right, so little of it is actually expressed. And some things that we thought in the past were considered genetic junk could be very regulatory in their functionality. Now, talking about now knowing that, let's look at biotechnology today. We've already talked about recombinant DNA technologies, but let's talk a little bit about CRISPR. And CRISPR is basically the ability to do gene editing. Now think about this, okay? Your DNA is your code book for you. And now there is a technology, and this is actually a very old technology, but new to us as far as our ability to harness it. And there are huge bioethical issues surrounding it, of course, and that is the ability to edit our own code. And who should be able to do that and what can be edited, right? That's a big That's a big deal. So CRISPR comes from clustered, regularly interspaced, short, palindromic repeats. Remember palindrome? It goes the same way, repeats same way forward and backwards, right? Like race car. Now, before I have already talked to you about, before in our video, I've talked to you about restriction endonucleases, right? Restriction enzymes, and that they cut at a specific series of bases, right? The restriction enzyme here is the Cas9. Those are the scissors. But where they're going to get cut, that guidance, like cut right here, which you can target, that is the guide RNA. And this, where you see it's tailor-made, that means that the guide that I associate with those scissors, I can pick that. So I can go have it find that particular of the DNA region. And not only can you edit it in the sense that you could cut out pieces of it or maybe insert a piece, but I could silence that gene so it could not be expressed, right? So that's a tailor-made sequence that ensures Cas9 is cutting in the right place or doing whatever else you want it to do. So you could modify the code, you could edit out genes, change the genes, the sequencing of it. Modify the histones. Remember the histones are where the DNA coils up on, and I could change what genes are expressed. Where did this technology come from? Same place as before, bacterial defense against bacteriophages that are invading them. So on your notes, let me help you fill that in a little bit. So CRISPR, the what? It is a tool for cutting DNA at a specific location. You can use it to repair DNA. That's little letter A. B, control of gene expression, turning genes off with CRISPR interference. That's where you put a little I at the end of CRISPR for sequence-specific control of gene expression. And then CRISPR-A would activate. It activates gene at a transcriptional level. So turning genes on and off. The discovery, I gave you that all in the notes about E. coli. The how. Your Cas9 complex consists of scissors that snips the DNA, and then your guide RNA, tailor-made sequence that ensures the Cas9 is cutting in the right place. Are there ethical issues? Absolutely, regarding the use of CRISPR technology, especially in humans, and those issues are not resolved yet. But it is definitely opening up huge avenues of study. Now, remember discovery, right? We want to always look at the bioethical issues associated with it. What I'm doing right now is just telling you about those. So next, what we need to do is, in our understanding of our human genome and all of the intra and intergenic sequences, we need to possibly expand the central dogma. Remember, the central dogma is that DNA is our code, right? And then you can get a copy of that code. RNA is the ambassador between the nucleus and the cytoplasm. And then you use that RNA to build your protein. And when we revisit our genetic, our central dogma, which is on your notes, you have expansion of the central dogma. Maybe we need to put more emphasis on the RNA that's generated. Maybe the product, right, is not the protein, maybe RNA and its regulatory function. Maybe that is the product. So more emphasis on the RNA generated, and a gene product may not be the protein. It may be the RNA. And then we also look at that some prokaryotes have RNA genes, and RNA could have come first, right, in our evolution. We've talked about that before. So on your notes, you have everything for that here on this screen to fill that in. So maybe put more emphasis on the RNA generated. A gene product may not be a protein, and some prokaryotes have RNA genes. So then going to functional and comparative genomics, with all this information, the aim is to understand the exact role of the genome. Before, we were just looking at the proteins that it would code for, and maybe there's some other functionalities that we haven't looked at yet, and trying to understand that exact role. And so put that in your notes. So one of those are DNA microarrays, trying to understand that. And what they've done is basically like a computer chip, and they have put sequences, base sequences, single-stranded DNA within these chips for, let's say, diseases. and then take your DNA and see if it is complementary to any of the DNA here. And that could indicate, that could give you a genetic profile of any diseases your DNA might be carrying. So DNA microarrays, DNA chips, current cellular, mRNA binds to DNA on a chip and can know which DNA is active, to know which DNA is active at that point, or what diseases you may carry. And that has everything to do with how it's complementary between DNA and DNA, or DNA and RNA, whatever you're using for your probeware there. A genetic profile can indicate genetic illnesses and what drug therapy might work best on you. And when you look at comparative genomics, comparing you and I and different groups of people, you can look at common features of different organisms, such as humans and fish are often encoded with the DNA evolutionary conserved between them. What are the similarities? So right here, you're looking at similarities between them. You can look at closely related species, such as humans and chimps, and show which genomic elements are unique. And then you can look at genetic differences within a single species, right? Such as our own, which can reveal variations, which may be leading to types of personalized, let me close this out real quick, types of personalized medicine. So look at your three different things here. Comparisons between completely different organisms, comparisons, excuse me, comparisons between similar organisms, and then comparisons within a single species. Guess I'm losing my voice this morning. All right. So on your notes, the word you need there is studying changes in evolutionary relationships. All right. And then this leads to the study of proteomics. And this is huge. And you know this because you know how proteins are built: primary, secondary, tertiary, quaternary structure, right? And tertiary structure is that folding pattern. Think about how critical those proteins are that actually do the folding to get it into the right shape. So proteomics is the study of the structure, the function, and the interactions of cellular proteins. And there are multiple goals, but here are two for you: to identify and determine the function of the proteins within the cells, and determine new, better, and individual-specific drug treatments. And I got this from a blog. You could just cut and paste this from this presentation. And I thought this was kind of a good way to look at it. After all, genomes are only recording information, right? Our DNA is a recording of that information, while proteins are the real ones that enact, right, what that cell is supposed to do and that activity. And you can use proteins for biomarkers, for cancer detection, antigens, or immune response, right? All the exchange in cellular communications. So for proteomics, you want to add in for your notes, the proteome is a collection of species proteins, which is larger than the genome. Now, why is it larger than our genome? Because you could have one gene, and depending on how you modify the mRNA, what introns and extrons you leave, right, that from one gene you can get multiple thousands, even millions, right, of different proteins just on how you edit that particular gene, right, when you form your mature mRNA. And studies the three-dimensional shape of the protein. And then I gave you these goals already in your notes. All right. And then bioinformatics. Bioinformatics is the application of computer technologies to the study biological information, looking at genomics, looking at proteomics. And so I gave you that in your definition. And I know some of you are like, duh, of course we're going to use a computer. But the massive amount of data that can be processed, one of those is BLAST, Basic Local Alignment Search Tool. That's what it stands for. And we will be doing a BLAST lab and using that computer technology when we look at evolutionary relationships. And what BLAST allows you to do is to compare homologous chromosomes between two different species, or to compare the proteins and look for similarity. What does that tell us, right? The more similar the genome, the more closely related we are. And so it shows you, it kind of breaks it down right here. Local means it searches or aligns sequence segments. It doesn't look at everything, just certain segments, right, to compare them, to detect relationships. And so it's a great analysis tool. We will be using this one right here from the U.S. National Library of Medicine, where we will be comparing and contrasting sequences of DNA and looking at evolutionary history. And if you're really interested in this right now, or those of you watching this video, I have a video just on that here on my YouTube channel. Let me pause and let me make sure I'll check the name of it real quick. Okay, so I just looked it up, and if you're searching this on my YouTube site, it's called APBio Investigation 3 Blast Lab, and I go through and explain how to do that lab if your teacher has already assigned that to you. And I also have another video where I show you, you can literally see me going through how to use that site. All right. And then that is the end, or is it? There's so much to go from here. We're just kind of scratching the surface. And if you're one of my students, I'll see you in class.